Hello and welcome to BIOS Frontier Science. I'm Kate Shannon, and today I'm excited to welcome Michael Levin, Professor of Biology at Tufts, Director of the Allen Discovery Center, and Associate Faculty at the Wyss Institute. To help with this episode, I'm also joined by my colleague, Chris Gabin. To kick things off, Mike, can you share a brief intro with us?
Sure. My undergraduate degrees are from Tufts University. I've got a bachelor's degree in biology and one in computer science. After that, I went to grad school. I did a PhD with Cliff Tabin at the Harvard Medical School in genetics. And after that, did a postdoc with Mark Mercola, also at Harvard Medical School, cell biology and developmental biology. And I started my own lab in 2000 at the Forsyth Institute, and I was there till about 2009. And then after that, moved my group to Tufts University on the Medford campus, and that's where we are now. My lab works on a number of issues related to information processing in cells and tissues and all kinds of unconventional media. So we work on basic questions of complexity and cognition and scaling of information processing, and try to apply that to practical things like regeneration, like machine learning, and so on.
Awesome. I'm excited to start diving into that. But before we get into the specifics of what your labs are working on, one question we like to ask our guests is, throughout your career, what has been your North Star or common thread that has tied your work together?
Well, conceptually, the thing that I was always interested in from basically as far back as I can remember being a kid, is this idea of the mind, or various kinds of minds inhabiting physical bodies. So looking at pieces of engineering, looking at bugs as a kid, and really thinking about how the behavior and the kind of the cognition of any given agent relates to what it's made of, how it got here, who created it, if it's an engineered sort of thing, and what kind of, what's it like to be certain kinds of agents, and what kind of information can they process about themselves, about the outside world. Everything we've done, even things that don't look like that at all, so all kinds of work in basic developmental biology, regenerative medicine, that don't really look like that's what they're about, that's what they're really about.
Amazing. I'm excited to hear more. And for our first section, I'll pass it off to Chris.
Thank you, Cage, and thank you, Professor Levin. So as you mentioned, at a high level, your lab focuses on biological information processing, on understanding the biophysical mechanisms that implement decision-making during complex biological pattern regulation. And you do this by harnessing the endogenous bioelectric dynamics that rationally control the growth and form of living organisms. You've also described this before as morphogenesis as a collective intelligence problem. So can you provide a high-level overview of those bioelectric dynamics and how they link to biological pattern recognition?
Yeah, well, let's take a step back and just get our bearings about the collective intelligence problem. So all intelligences are collective intelligences. People often will think that, well, there are collective intelligences like ant colonies and beehives and things like this, and then there are real intelligences, in a brain and so on. And all intelligences are made of parts. So we often feel like a coherent, integrated, single agent that has consistent goals and preferences and memories and so on. But we're all made of cells. We're all collections of cells. And it's a fundamentally important question to ask: how do collections of things, whether they be cells or robots in terms of swarm robotics and things like that, how do collections of competent subunits work together to make something that's more than the sum of its parts, that has unique memories, preferences, goals, and behaviors that don't belong to any of the individual components? And that can take place, that kind of activity can take place in lots of settings. It can take place in familiar three-dimensional space, such as when ants work together to maintain a hive and so on. But it can also work in other spaces like anatomical morphospace. So when you think about how it is that embryos self-assemble, or why do certain types of animals, let's say salamanders, regenerate their limbs, it all boils down to this collective intelligence problem of, you have a group of cells. Cells are individually competent to do various things. How is it that they work together towards specific goals? Now, how do you know they work towards specific goals? Because you can deviate them, you can do all kinds of things. And as far as they're able, they will, for example, regenerate a limb, and then they stop when it's done. That's the most amazing thing about regeneration, is that it knows when to stop. So there's a very particular goal state, and then when that's achieved, then they stop. So we're interested in development, and I went into biology. I started out in computer science because I was interested in artificial intelligence and making novel kinds of minds and different implementations. But it became clearly, sort of very clear early on that biology has some amazing examples of that process of scaling individual cell activity, right? We all started life as one cell, and somehow that process not only gave us our bodies, but actually our minds as well. And this is a fascinating set of questions. And the way you get to bioelectricity from that is by asking, okay, so you have all these individual agents, they have to communicate. There has to be a medium that holds the cognitive content of the group, right? So if you're going to create a new whole out of pieces, where is the cognition stored and processed of this new individual that appears? And in neuroscience tells us very clearly that it's in the electrochemical networks of the brain, right, and the nervous system. And in fact, it turns out that evolution discovered this, the fact that electricity is a great way to scale tiny little goals of individual cells into very large scale goals of making body types and so on. It turns out that evolution discovered this very early on. So even bacterial biofilms do some of this, and long before bioelectricity became useful for controlling movement in three-dimensional space, it was used to control events in morphospace and anatomical remodeling. And so this is why I became very interested in bioelectricity. My lab created some of the, there's a long history of people interested in the problem of bioelectricity, but generally not from a cognitive perspective. Generally, people look at it as another piece of physics that's important for cells to do what they need to do. I think that's all true, and there's an amazing amount of work on this from past workers in the field. But there's a new component to this, I think, that is only now beginning to be appreciated, which is that it's not just more physics. It's actually the information medium in which that collective cognition operates. And so we developed some of the first tools to molecularly track this process. So to detect voltage gradients across living tissue, and then so that's reading that information, and then to rewrite that information by altering the function of the channels and pumps that give rise to these gradients.
Before diving in a little more to those tools, you've talked about how, as a pioneer in this field, you're really crafting that understanding of collective intelligence. Can you share more about some of the key interdisciplinary aspects and what first led you to bring them together?
Yeah, and I want to be really clear. There are lots of people who have done amazing work that relates to this, and I'm for sure not the first person to be interested in these questions. I like to think that my group puts them together in novel ways, but there are many really interesting labs that work in collective cognition, in swarm intelligence, and so on. What led me to think about it this way? Again, my background being computer science is all about understanding how to build things. It's a kind of engineering, right? Where you don't, you don't really understand something until you can build one yourself, basically. But at the same time, one of the most powerful concepts in computer science is device independence. The idea that you can carry out an algorithm and it's the same computation, whether it's made of microchips or beer cans and string or living cells or whatever it's going to be. This is an important concept, and it's really it's a tough one for a lot of biologists to sort of think about it this way. That we biologists are really used to thinking that everything is in the details. You have to know exactly which protein it is, which gene it is. The details are what matters. And computer scientists take a slightly different approach, which is, let's look at all the stuff we could swap out and still have the heart of the process. And so that's what I started out by thinking, that if that's true, we can really look for invariance in all these deep ideas, all the things coming from neuroscience about memory, about preferences, about goals, about having the boundaries of the self versus the outside world, the computations. It really then let's look at all the things that are essential to this and all the things that are not essential. And I would argue that what's not essential is how you got here, meaning whether are you evolved or designed. right? Because that in the end, that doesn't matter. And what you're made of, because the same dynamics can be executed in lots of different media. And so I think the biological world is amazing at being able to teach us what's possible, but it's an N of one example, right? This is how biology evolved on Earth. This is a bunch of frozen accidents that sort of, determined how life advanced here. And this is why I'm very interested in synthetic morphology and artificial life, in really broadening it out, as Chris Langton had said, life as it can be, not just life as it is.
As we start to think about that life as it can be, and as you touched on before, while investigating this space, your lab has developed a number of next generation tools to help scientists understand that sort of top-down control of pattern regulation. So can you start by telling us a little bit more about the interplay, and you've begun to touch on this, between biology and machine learning?
Yeah, so the relationship between biology and machine learning is kind of a back and forth feedback loop. So we are interested in using techniques of machine learning to help us understand what the biology is doing and to uncover patterns that we wouldn't otherwise have seen, because we're not ready for them, and our perceptual and cognitive systems aren't built to understand, for example, movement through other spaces that are transcriptional space or physiological space that are not three-dimensional space. So I think machine learning is very useful to help us understand some of these things. At the same time, I think that many of the things that we discover in the end are going to feed back and enrich areas of machine learning and computer science. First of all, by teaching us about the benefits of this multi-scale competency architecture that we see in life, where every piece of that nested doll that we are, not only are we made of modules, but what's important is that every module is a problem-solving basal intelligence that has some capacity to pursue certain goals in relevant spaces. And that's a very powerful architecture that evolution discovered that we don't exploit really yet in robotics and so on. So it's sort of, and there's another aspect to this, which is that, can we make machine learning architectures that don't rely on a brain like sort of, on a neuromorphic kind of architecture, right? Can we exploit the way that life has been solving problems for a really long time before brains showed up on the scene? So I think there's that kind of back and forth between machine learning and biology.
And so given that overview, can you tell us a little bit more about the tools your lab has developed and how they've been progressing your research in the field?
Yeah, we've ended up sort of borrowing a lot of things from neuroscience, and it's not an accident. It's one of the interesting things in this whole field is that people often think that it's very easy to know what a neuron is, and it's actually not obvious at all, because most cells in the body do the same kinds of things that neurons do. They all have ion channels. They all have voltage gradients. Lots of cells use serotonin and other neurotransmitters. They have electrical synapses that they communicate with. And the way that on the one hand, it's kind of weird to think about other cells in the body having these kinds of networks, electrical networks that you expect in the brain. At the same time, if we take evolution seriously, what else would we expect, right? Neurons didn't just appear out of nowhere. They sort of speed optimized things that lots of cells were doing before nervous systems came along. And in fact, because of this, all of the techniques of neuroscience can't really tell if you're dealing with neurons or not. So optogenetics, the use of drugs to target ion channels and neurotransmitter pathways, voltage sensitive dyes that can be used to track electrical potentials, all of that stuff carries over very nicely. And in fact, I've made the case that I think not only the techniques carry over, but actually the deep insights of neuroscience, all of that multi-scale activity of trying to build up all the way up through psychological, kinds of propensities from lower levels of electrical network activity and individual synaptic activity and individual ion channel activity and so on. All of that is directly importable from neuroscience. So basically, what we do is we use voltage sensitive fluorescent dyes to track the electrical communications between cells and all the electrical states in tissues during regeneration, during development, during cancer. And then we use the same tools to perturb those electrical states. So we can mutate ion channels. We can use drugs to open and close ion channels. We can use optogenetics to lay down light masks to control electrical activity. What we don't do is any kind of, there's no electromagnetics, there's no magnetic field application, there's no electrodes, there's no electric fields. It's all done with molecular pharmacology, basically targeting the native computational interface of cells. All cells expose this amazing interface, these ion channels on their surface, and that's there for the system itself, and then for us to be able to communicate with them.
So your lab has advanced our understanding, and as you touched on, how somatic cells from bioelectric networks store and recall these memory patterns that guide morphogenesis. Can you give us an overview of how the biological process works?
Yeah, so I'll just give you a simple example. There's many, but one cool one is called the electric phase. So this was discovered by my colleague Danny Adams when she was a postdoc in my group, and what we were doing was using voltage dyes to study the electrical gradients in the ectoderm of the early frog embryo as it's starting to put its face together. So what we found out is that if you we have videos of this on our website, that before the genes come on that regionalize the face and dictate where the mouth and the nose and the gills and the placodes and everything else is going to form, there's a very particular bioelectrical pattern that is basically a scaffold or a pre-pattern that's going to direct where the genes become expressed and where the anatomy is going to go. And so this electrical pattern is basically a readout. And this one happens to be pretty direct, which is one of the ones we discovered first because you can sort of see it. It just looks like a face. There are other patterns that are much more difficult to interpret. They don't look like what they really are. They have to be deconvolved. But this one basically looks like a face. And so what happens is that these patterns, these electrical patterns self-organize in a field of cells by symmetry breaking and then amplification. And you can think Turing patterns is a way to think about this if it were a chemical medium. And then the bioelectric circuit stabilizes and it regionalizes different domains of the tissue into different voltage states. And then those patterns kickstart different downstream cellular activities. And this is called, we call this the bioelectric code, which is the mapping between the bioelectric pattern and what happens downstream. Now, the voltage distributions kickstart, they get transduced into second messenger cascades. So we're talking about movements of calcium, of neurotransmitters like serotonin, clustering of KRAS in the membrane, of changes in the cytoskeleton, voltage sensitive phosphatases. There's probably about half a dozen known ways that bioelectric patterns get transduced to downstream events. And the important thing about these patterns is that they're not single cell events. So it's not, the question is never what voltage is any one particular cell. The question is what's the large scale pattern on a field of cells. And those patterns code for specific outcomes: build an eye, build a leg, build a heart, a brain, and so on. They demarcate the edges of different compartments, different developmental compartments. And so when that happens, then the conventional machinery takes over. The genes get turned on, and then the whole thing is a big cycle, right? Because those events can also change the way that ion channels operate. New ion channel genes might become expressed, channels might be open or closed, and the whole thing sort of rolls on as this dynamical system.
Passing that back to Cage, I'd love to hear how this all ties into what might be called the future of medicine.
So as we think about innovative research, we also like to consider potential applications for human health. So tying some of your work together, how does your research apply to the future of medicine?
Yeah, so a couple of things to think about. The first thing is that oftentimes people will ask, well, you've shown a lot of, and other people have shown a lot of data on bioelectrics in flatworms and frogs and things like this. How do we know that it's even relevant to human health? And the important thing to keep in mind is that this kind of mechanism is incredibly well evolutionarily conserved. Human channelopathies tell you that this is relevant to humans because mutations in ion channels, for example, in human patients, lead to exactly the same dysmorphias, developmental dysmorphias, as you see in the animal models. Now, and so all cells are tuned to these bioelectrical control pathways. Now, what we hope to do is build long-term. The vision is to build machine learning platforms that help to do two things. to help you build a circuit model of what type of electrical activity is responsible for the correct bioelectric state that drives healthy morphogenesis. And once you have that model, to be able to ask that model questions like, well, if the pattern goes wrong due to disease or injury or exposure to teratogens or something else, what channels would you have to open and close to get you back to the correct state? Because if you can do that, then you can go to the shelf and find one of many ion channel drugs that are already human approved. And this opens up a whole class of electroceuticals of human approved ion channel drugs that can be repurposed for guiding these bioelectrical gradients. And we see applications, I mean, we can dive into the details if you want, but we see applications in birth defects, in normalization of cancer, and in regenerating damaged organs.
Interesting. I think we'll dive a little deeper in a second, but how does this approach differ from traditional therapeutics?
There's two ways. There's the short, sort of the short-term view and the long-term view. In the short term, what we have is we're targeting a different aspect of cellular control. So whereas most people might be targeting transcription factors or proteins that operate various kinds of pathways, we are targeting a different level of the physics and physiology. We're targeting the ion channels and the electrical machinery that stores some of the information that cells need to know in order to know what are they working on, what are they building, repairing, and maintaining. That is somewhat different because you have to think quite differently about this. For example, in traditional molecular medicine, if you have a specific gene that does a specific job, a specific protein, that is the gene you need to worry about. You might upregulate it, downregulate it, mutate it, whatever. In bioelectrics, the driver is the electric pattern, and the specific ion channels that created that pattern aren't that critical. I mean, they're important as far as being control knobs for you to impact during interventions, but the magic isn't in the genes themselves. And this is one of the things that is really hard to get your head around, because I'll just give you a simple example. We did some work on tail regeneration. Okay, and it's important because it has spinal cord and all that. And so in the tadpole, the tail, in order to regenerate, requires a particular type of a proton flux that's carried out by this particular proton pump. And we showed that if you shut down that proton pump, then the tail fails to regenerate. But you can rescue the whole thing with a proton pump from yeast that has absolutely no sequence or structural homology to the original protein. So this is now, right? And so this shows you that the identity of the protein, the networks that build the rest of the tail that need to be pushed into the build a tail program as opposed to make a scar program, they don't care at all what gene or what protein generated the voltage flux. They are triggered by a very specific set of electrical parameters. And if you can get there with potassium or sodium or chloride or protons, they don't know it and they don't care. It's the voltage pattern that's important. And so that means, the good news is that means you're free to swap out whatever's convenient, right? You're free to, it's not the specific gene that matters. It's let's just get the voltage right by opening and closing whatever, and it doesn't matter what the native trigger was. That's kind of the short-term difference. The long-term difference is deeper than that. Right now, with a few exceptions, most of our I think this is true. Most of our interventions in medicine address specific symptoms. In other words, most things don't actually cure anything, right? Most things you take the drug, hopefully the side effects are worth the benefit, and then when you stop taking the drug, you're back where you started from, right? So it's a design, and the reason is because it's not designed to fix anything. It's designed to address the state, it micromanages the process, right? You see what's wrong and you try to fix that particular thing. That level of, and that approach works great for certain things, but for very complex things like the process of regenerating a limb or the process of restoring physiology or maybe aging and things like this, that micromanagement approach is not going to work. It is just the complexity barrier is too high. And in those cases, what I think you would need to do is take a step back and ask, what information is the system processing? What memories, what goals in various spaces does it have that we could exploit, not micromanage, but exploit in terms of the native sort of software that the system already has, that we can control it at a much higher level. I'll just give you a very simple example from neuroscience. If you have a rat and you want the rat to do a circus trick, put a little ping pong ball in the hoop, right? You got two choices. You can try to micromanage the process and control every single neuron that might be involved in getting its body to move. So you'd have some sort of a controller that works the rat like a puppet and tries to trigger every particular neuron. I mean, probably that's possible, but how many years, how many decades before we're going to be able to do that? On the other hand, people have been training animals for thousands of years, knowing nothing about what's between their ears. You can just train the rat, and you know that won't take you very long, and the performance will be great. And the reason that works is because what you've realized is that the rat is a learning agent. It's not a puppet. It's an agent that has preferences and learning capacity. And you figured out what the top level triggers are to get it to do what you want it to do, right? You figured out the currency of incentivization for the system. There's certain things it likes, certain things it doesn't like, and you don't have. And so the benefit of knowing that is that you can now work at a very high level. You can specify your goals you can work in reward space as opposed to the unbelievably large space of every possible neural activity that you would have to control. And so now that kind of approach, it saves you a lot of time and effort, but it works with rats. It doesn't work with mechanical clocks. You're not going to train a clock. And so the key then is to look at your biological system and ask, where on that spectrum from a completely mechanical contrivance to one that has some learning ability, some built-in software that you could take advantage of, where are these biological systems? The assumption in modern medicine is that they're all on the side of the clock. Everything is a clockwork mechanism. We're going to rewire pathways. We're going to re-engineer them with synthetic biology, with genomic editing. This is, if you think about computer science, this is how programming was done in the 40s and 50s, right? I have this picture that I show in my talks of a woman programming a computer in the 1940s. What is she doing? She's physically replugging the wires back and forth, right? Because that was the only way to do it. You had to interact with the hardware. Nowadays, nobody thinks twice about the amazing magic of the fact that when you, on your laptop, when you go from Microsoft Word to Photoshop, you don't get out your soldering iron and start rewiring the microchips. Well, why don't you have to do that, right? I mean, the hardware is really important, because if the hardware is good enough, you can control it with inputs, with stimuli, with experiences, not by rewiring. And so the real question is what kind of biological problems are amenable to this latter strategy and not the former? And so this is what my group is very interested in, is looking for signs of what's called basal cognition, which is very simple kinds of precursors to human advanced cognition that have to do with motivation. Can we train cells to do particular things? Can we incentivize them with rewards, with various things that cells like, to have specific morphogenetic behaviors, to build an arm, to do things that we don't know how to micromanage them to do? And we found we had a paper recently showing that even models of gene regulatory networks, which are like the most basic sort of deterministic, hardwired kind of a thing. I mean, you can see there's no magic. You can see all of it, all the genes turning each other on and off. Even those things are capable of associative learning. And I think that this kind of stuff is sort of conserved all the way down to the tiniest pieces of the biology. And so I think the future of all this is to use the bioelectrics not the way that bioengineers currently want to use it, which is just to push individual cells to do specific things. People often ask, can you use bioelectrics to turn a stem cell into a whatever cell type? Probably, but I don't think that's the most exciting part. The most exciting part is that the bioelectrics gives you access to that interface, to that software interface. Every cell exposes these ion channels on their surface as a way for you to communicate with it, not to try to force it to do things. And so that is I think that is going to be the future of biomedicine, where we incentivize, reward, motivate, and otherwise take advantage of the native plasticity that these collective intelligences have. That, I think, is the big advance going to be.
So as you bring forward this new approach and really this new way of thinking about biology, what do you think the best or most interesting applications are going to be?
Yeah, I think the three areas that I can think of right off the bat. First is birth defects, and we've already shown some interesting examples of this, of being able to repair extremely complicated, nasty birth defects of the brain induced by mutations of genes like Notch, critical neurogenesis gene. You can get a normal brain back with normal structure, normal function, and normal behavior. I mean, they get their IQs back despite these mutations, despite exposure to alcohol and nicotine and all this stuff, because we had a computational model that reveals what the correct, how the correct bioelectric pattern can be restored from an incorrect pattern, right? And we were able to do that with ion channel drug exposure that was predicted directly by the model. So that's an example. Regeneration is another example, and I have to do a disclosure, because David Kaplan and I have a spinoff called Morphoceuticals Inc., which is around taking what we've learned about inducing frog leg regeneration and moving it forward into mammals, and then hopefully someday human patients, to try to trigger the regeneration of lost appendages, fingers, limbs, and so on. And then the third one is cancer. So this is a pretty natural fit, because if you think about what really goes wrong in cancer, is what's the biggest, and cancer of course is very heterogeneous, but the biggest sort of central thing is that cells revert back to their unicellular goals being very small, right? Back in the day when we were all amoebas, your goals were tiny. What did you want? You wanted to be, every cell wanted to be 2 cells and go where life is good. That's it. And only later, when they started working together, this cognitive scaling took place, where they're able to work on very large goals. Hey, let's make an arm. And by the way, a bunch of us are going to die through apoptosis, but that's okay, because we're going to achieve this arm, right? And so that scaling of the goal, well, that can shrink again. And individual cells that express oncogenes that cause them to electrically decouple from the rest of their neighbors, at that point, their cognitive horizon shrinks. The rest of the animal is just environment to them. That boundary between me and world, it might be around the size of the organ or the whole animal, but once you go convert to carcinogenesis, it just shrinks, and now you're one cell again. And so we've shown again in the frog model that you can trigger cancer with no DNA damage, no carcinogens, just by decoupling cells from their neighbors. And better yet, you can do the opposite, which is you can put in oncogenes that normally cause tumors and suppress that process, and you don't kill the cells, they just basically normalize and they become normal cells by carefully managing the electrical state and their connection to their neighbors. So I think those 3 areas, birth defects, regeneration, and cancer, are some of the best application areas for this.
Those are some fascinating applications, and we'll definitely look forward to watching those progress. But for now, I'd like to shift tracks just a little bit, talk about synthetic biology, which has been rapidly gaining momentum in recent years. And with some saying it heralds the 4th industrial revolution. You've described synthetic biology as the plasticity of life. Can you explain what this phrase means to you?
Yeah. One of the most interesting things about life is that at all scales, living things solve problems in different spaces. And in particular, what I mean by plasticity is that they handle novel circumstances. So we have lots of examples of biology just being amazing at reaching the same outcome despite radically changing circumstances. Now that, by the way, is William James had this definition of intelligence as the ability to get to your goals by varying means, right? That's a kind of intelligence. So just to give you a couple of simple examples, this is very old. This one's very old. Salamander newt kidney tubules. If you take a cross section, it's usually a bunch of cells sort of working together to make this nice ring of cells that make a tubule. If you make a polyploid newts, which have more DNA than they're supposed to, the cells get bigger. The overall newt stays the same size. So what that means is that fewer cells get together and build the same exact kind of tubule. The kicker is that if you make really huge cells, one cell will bend around itself, leaving space in the middle, and make your tubule. Now, what that is completely different molecular mechanisms being harnessed and called up to in the service of an anatomical goal state, right? Well, we don't have any molecular models that even begin to explain any of this, but It's an amazing degree of plasticity because we don't have any technology that can, hey, by the way, I just made all your parts 5 times larger. No problem. I've still made, I still made a bicycle. We can't even, like, we don't have anything that does this. Our computers can't do it. Our robotics doesn't do it. So that's one example. Another example is, and this one we discovered, typical tadpoles to turn into frogs, their face has to rearrange. So the jaws have to move, the eyes have to move, the nostril, everything moves. And it was assumed that somehow that basically what you had was a set of hardwired movements, because every tadpole looks the same. So if you just take a standard, if all the organs move in the right direction, the right amount, then you get your frog, right? So by disturbing that electric face that I showed you, we were able to make these Picasso tadpoles. And these Picasso tadpoles, everything is in the wrong place. So the eyes on the back of the head, the nostrils are off to the side, the jaws are, everything is just moved around. Well, guess what? They make largely perfectly normal frogs, because all of these things will continue to move in novel paths. And in fact, sometimes they go too far enough to double back, and they keep doing this until they end up in the correct configuration to be a frog face. So what the genetics actually specifies is an amazing machine that has the ability to minimize error. It will continue to act until the error, the delta between the correct frog face and the current frog face, is as small as it can get. And so these types of, this is what I mean by plasticity, is the ability to still get your job done. Think about what that means for evolution, right? It means that if you have a mutation, I mean, it just really cranks up evolution in an amazing way, because if you have a mutation that moves your eye and at the same time does something good in the rest of your body, normally, if your eye now doesn't connect and you have no vision, well, you're dead, and evolution would never see the benefit of that mutation. But if your eye will just find where it needs to go, now you have the ability to explore this other positive effects of that mutation, right? Because the eye will find its spot eventually. So that kind of plasticity is incredible. And so for synthetic biology, what it means, I think, is that, and our efforts on synthetic morphology, which are these Xenobots and so on, that really what I think we're going to be able to do is take advantage of all that. So when you're doing synthetic biology, yes, you can put in new things that have never been, that the system never did before, new circuits and new metabolic cascades and so on. But what you ought to also could do is take advantage of all the things that, of the plasticity of the system, that you don't have to micromanage. The fact that cells and tissues are really good at novelty, at solving novel problems. And we have lots of examples of them solving physiological problems and so on. You could take advantage of all that, which makes your job as a synthetic biologist, on the one hand, much easier because you don't have to build everything from scratch. It also makes things harder because there's going to be lots of surprise. You're going to try to do things and the system's going to get around whatever you were trying to do. So it's a very, super exciting time.
Awesome. And so in that last response, you mentioned Xenobots, which in my opinion is one of the coolest applications in recent years that your lab has been working on. The first one coming from frog stem cell assemblies in 2020, and the publication announcing the second generation of self-replicating Xenobots being released just last Monday. Can you tell us a little more about your work with Xenobots, what led to their design, and kind of where are they today?
Sure. Yeah. And so this is a project that's done together with our partners. So this is the lab of Josh Bongard at the University of Vermont, and then Sam Kriegman, he was in Josh's lab as a PhD student. He's now a postdoc in my group, and Doug Blackiston, who's a staff scientist in my lab, who was responsible for lots of interesting advances that we've made. The big thing about Xenobots, I'll introduce it this way. What we wanted to do was we wanted to investigate this plasticity, and we said, okay, if we take some cells out of their normal context of an embryo, we basically liberate them from the rest of the embryo. We don't add anything. We don't give new transgenes. We don't edit the DNA. We subtract some stuff. We subtract the instructive control that the other cells had over them. Okay, and we set them aside in a novel environment, and we basically let them reboot their multicellularity. Right, you're on your own now. What are you going to do? And many things were possible, right? They could have formed a monolayer on the bottom of the dish. They could have crawled off and separated from each other. They could have died. There's lots of things they could have done. What they instead do is they come together within about 48 hours. They come together into a novel proto-organism consisting of just skin. There's no brain. There's no nervous system. It's just skin that has a bunch of different capacities. And the capacities it has are because it repurposes the genetically provided hardware, for example, cilia, the little hairs that normally wave and sort of push the mucus down to the bottom of the tadpole. Now it uses them to row and to swim against the water. So Xenobots, it turns out they can swim. They get around. You can put them in a maze, and they sort of get around. They're self-actuated. You don't need to pulse them with anything or to make them move. They go on their own. They self-heal. If you cut them almost in half, they will sort of rejoin and try to get back to their Xenobot shape. They have all kinds of interactions with themselves and their environment. And what we showed in our latest paper this week is that being deprived from the ability to reproduce in the normal way, they figured out a new way to self-replicate that is, to our knowledge, completely unique, not only to the frog lineage, but anywhere. I don't know of any animals that do this. They can basically run around and collect other cells and push them together into piles and make copies of Xenobots themselves. And this goes on for multiple generations. And so I think there's a couple of interesting implications here. One is, sort of foundational, which is that where do these things come from? In the sense that for any other creature on Earth, if you were to ask, why does it have a certain number of fingers? Why is it this color? Why does it jump or fly? The answer is always the same, because for millions of years, its ancestors were selected for the ability to do X, Y, Z. Xenobots never existed before. They figured this out in 48 hours, how to repurpose what they have into an entirely new functionality. Where does that come from? We have absolutely no idea. And that has massive implications, I think, for our understanding of evolution, of the origin of novelty in biology, and actually for dealing with a lot of systems that we're now making, swarm robotics and things like this, to understand where the goals of collective intelligence has come from, right? Anatomical goals, behavioral goals. So that's all sort of theoretical interest. The practicalities are twofold. One is that they can, the more we figure out how to stimulate these cells to build what we want them to build, so different kinds of bots and so on, the better we get at making useful synthetic living machines. So we're talking about, microsculpting things for transplantation, cleaning out biofouling, various kinds of environmental cleanup and sensing operations, exploration and rescue inside the human body, chasing down cancer cells and fixing wounds and so on. That's all sort of one set. The other set of longer term applications is that these Xenobots are the first of a class of technologies that will help us crack the morphogenetic code. This question of how do we control the states towards which cellular collectives will work? We have no idea how to do that. So this is a kind of a sandbox platform to figure out how do we communicate to a cellular collective. This is what I want you to do. I want you to build a heart or a liver or an arm or an eye or a different eye that I'd like to have that no one has ever had before, whatever. So that kind of a thing. It's a discovery platform to crack the morphogenetic code.
So you've led perfectly into my next question, which is, as you look forward, are there any applications for these Xenobots that your lab is considering? And projecting even farther forward, how do Xenobots tie into the future of synthetic living organisms?
Yeah, I think Xenobots are one example of synthetic living organisms. I think there will be many other examples. I think already, people often will say things like, well, the human being this and that, and and robots are whatever. There has been so much overlap already. People that have been instrumentized with insulin pumps and things that allow them to control their wheelchairs via their brain activity through a machine learning interface. Like the barrier between all these types of life forms is gone. And in I think in the next couple of decades, we're going to see an incredible proliferation of hybrids, cyborgs, every possible kind of novel way of being alive and cognitive and so on, both very, very primitive things like Xenobots and probably very advanced things that are combinations of humans with other technologies, and everything in between, every step in between. And so this is just the beginning. This is telling us the discovery of this kind of plasticity of cells is reminding us that the life we see around us is just one example. and that there's much, we're just a tiny corner of the possible space of different ways to be an agent.
Amazing. This has been an absolutely fantastic discussion. But before we go, I'm going to pass it back to Chris for just a few last-minute questions.
Thank you, Cage and Professor Levin. A few rapid-fire things to cap our conversation. First and foremost, do you have any advice for early career individuals hoping to explore or pioneer new fields?
Well, that's a lengthy topic. I think the short advice I can give is this: read very, very broadly, and do your best to prevent the frameworks of any one field from constricting the way you think about things. What I've noticed throughout my years of working on all this stuff is that things that are incredibly obvious in one audience are a complete taboo somewhere else. And the way that different fields think about things is often extremely constricting. So beware of, you want to, I mean, it's hard. You want to pick up the ways of thinking of particular communities because they're helpful, but along with that come blinders that keep you from seeing things in new ways. And we have a real problem with silos in science where certain communities are just very, very close-minded about certain things because that's how it's always been for them. And so if you're young and you're entering this field, lots of people will tell you, that's impossible and this can't be done and that's not a robot and all of these things are crumbling. Okay, that we're going to have to rebuild this basically from scratch. These distinctions between sciences come from a time where we really didn't know anything, and we still don't know anything. But one thing I think we do know is that these distinctions are contingent and not deep. And so read broadly in different fields, get lots of interdisciplinary training, and try not to take on the limitations of any one field.
Jumping off that inspiring message, do you have any other closing thoughts or shameless plugs?
No, all the details are on our website. Just come to drmichaellevin.org and download anything you want. We have software, protocols, reprints, everything.
Great. Thank you, Mike, for an absolutely incredible episode. I'm sure our listeners had just as much fun as we did. We're very grateful for your time. Thank you again.
Cool. Thanks very much. Yeah, it was nice to talk to you. Thank you.